Good morning, viewer. Welcome to our FTV. Viewers, Mayo Clinic researchers have identified an immune regulating molecule that may help explain why some patients with inflammatory bowel disease in the short form IBD, including Crohn's disease and ulcerative colitis, do not respond to commonly used therapies. The findings published in Cell Reports described a previously uncharacterized role for the molecule ST8-SIA6 in regulating immune activity in the gut. In preclinical models, researchers found that the absence of ST8-SIA6 led to a marked increase in inflammatory immune cells in the intestines. The results suggest the molecule plays a key role in maintaining immune balance and may represent a distinct biological pathway from those targeted by existing treatments. Viewers, IBD affects nearly 3 million people in the United States and its prevalence continues to rise. While the source of IBD is not known, researchers believe it may occur due to a combination of genetic and environmental factors. Recently introduced medications target a pro-inflammatory molecule known as TNF alpha to reduce the autoimmune response in the intestine and help with symptoms such as diarrhea, blotting and bloody stools, but the treatment is not effective in all patients. Discovering the role of the enzyme ST8 SIA6 has provided the research team with a new view of IBD. The normal function of ST8 SIA6 in the gut had not been previously described, says Mayo Clinic immunology researcher Virginia Safiro, PhD, principal investigator of the study. We found ST8 SIA6 regulates the abundance of immune cells and keeps them in the steady state of homeostasis. When the molecule is not present or even is reduced, the presence of inflammatory immune cells increases dramatically. The study of ST8-SIA6 and its interactions with the immune system extend recent discoveries in Dr. Sapri's research lab. Her team previously slowed the ST8-SIA6, an enzyme that adds sugar molecules to cell surfaces, enables a tumor to evade destruction by the immune system. The lab also found that ST8-SIA6 can be used as a tool to help reduce the body's autoimmune attack on insulin producing beta cells as occurs in diabetes. The leap of gastrointestinal disease occurred when the team noticed on an international database that a single mutation in the gene for ST8-SIA6 turns up with greater frequency in people with Crohn's disease. Viewers, the ST8-SIA6 molecule is expressed pretty widely in different immune cells, but we were not sure of its connections to this disease, which involves chronic inflammation, says Sidney Crotts, a graduate student at Mayo Clinic, Graduate School of Biomedical Sciences and first author of the study. The team looked at preclinical models that lacked the ST8-SIA6 gene in the molecules and abundance of immune cells gathered in the small intestine. The team also found that lower ST8-SIA6 led to increased levels of messenger molecules that prompt an immune response. So, together the attributes increase susceptibility to intestinal inflammation. We think this might be a model of what's happening in patients with Crohn's disease who have a baseline of immune cells and are basically fine until they encounter a trigger and have a flare, says Crotts. Say notes that the inflammations did not abate with TNF alpha medication, suggesting this pathway may be distinct. Bravo, sir. Bow down. We have this exclusive report, FTV English, Delhi, Bharat.